Hey everyone, welcome back to my channel. So today I will be talking about the structure and function of messenger RNA, transfer RNA, and ribosomal RNA. And before we talk about these three major types of RNAs, I first want to quickly touch base on the structure of RNA and how it is different than DNA. So RNA, which is ribonucleic acid, it contains ribonucleotide, and this ribonucleotide is nothing but it's just a nitrogenous base plus a sugar molecule. So in RNA the sugar molecule is ribose sugar, whereas in DNA the sugar molecule is deoxyribose sugar, as well as it also contains phosphate molecule. So this is a ribonucleotide, which is nitrogenous base, sugar molecule, and a phosphate molecule. And it is also called as nucleoside monophosphate because nucleoside is equal to nitrogenous base plus a sugar molecule, and monophosphate because it is attached to one phosphate molecule. So, and when this nucleoside is attached to a phosphate molecule, it becomes nucleotide. And you can review this in detail in my previous video of introduction to nucleic acid, nucleoside, and nucleotide. So now basically compared to DNA, RNA is very small, whereas DNA is very large molecule. And as I mentioned, it contains ribose sugar instead of deoxyribose sugar of DNA. And the another major difference is RNA has a pyrimidine nitrogenous base which contains uracil instead of thymine, which is a pyrimidine nitrogenous base of DNA. And unlike DNA, RNA exists as a single strand molecule and capable of folding into complex structure, in which we are going to talk about which we are going to see in subsequent slides. So unlike DNA, because DNA is a double-stranded molecule. Now RNA is also important in transforming the genetic information because it can transfer the genetic information which is stored in DNA, and that genetic information then will be utilized to synthesize protein. Now they are located outside the nucleus, unlike DNA, which is located inside the nucleus. And compared to DNA, they are inherently very unstable, and which we'll see why. And like I mentioned, the most important function of RNA is to participate in protein synthesis process. So this diagram here is very helpful to understand the entire process. So here you have your DNA molecule, which is located inside the nucleus, and the genetic information DNA is transcribed into mRNA, which is synthesized in the nucleus. So the transfer of genetic information from DNA to RNA is called transcription. And once the genetic information is transcribed into RNA, RNA is then translocated into the cytosol, and this the genetic information from mRNA is then used to synthesize proteins, and that the process of that is called translation. So now there are three major types of RNAs. First is messenger RNA, second is transfer RNA, and third is ribosomal RNA. So we're going to talk about this individual RNA in detail. So now let's first talk about messenger RNA. Now messenger RNA, it represents only 1 to 5% of total cellular RNA. However, it represents 50% of total RNA synthesis, and messenger RNAs are very important as it carries the genetic information from DNA, and that genetic information can be utilized to synthesize protein. Now they are most heterogeneous in size and base sequences, Therefore, it can also be 1,000 base pair long molecule. And I have been saying this further again and again, that it carries the genetic information from nuclear DNA, because DNA is located in the nucleus. So from nuclear DNA to the ribosome. So once it carries the information, it translocated into the cytosol, where to the ribosome, which is located in the cytosol. And ribosome is also the site of protein synthesis. So, and then that process is called translation. And they are very short-lived. They are very vulnerable to degradation, and we'll see in the structure compared to DNA, they are very short-lived because they are very prone to degradation in the cytosol. And most importantly, mRNA encodes for protein synthesis. So genetic material which is transcribed from DNA, that is coded for protein synthesis. So this is the basic structure of mRNA. So here at the 5' terminal, you have this cap structure. This is, I call it as a methyl cap. So this is actually, once the mRNA is synthesized in the nucleus, on the 5' terminal end, there is 7-methylguanosine which is added. I kind of call it as a methyl cap, and once this methyl cap is added, it's kind of an indication that it is now ready to transfer into the cytosol. So on the 5' terminal end, we have a methyl cap, and this is a coding region. So in this middle part of the mRNA, which is in blue, this is called a coding region. It's called coding region because it encodes for protein synthesis. So this is a very important region. And right before the coding region, we have 5' UTR. Now UTR is called untranslated region. So this coding region encodes for protein, and this 5' UTR, as the name suggests, untranslated region, because it doesn't code for anything. And the most important function of 5' UTR is, once the mRNA is translocated into the cytosol, it indicates for the translation initiation sequence. So this is important to initiate the translation process. Now on the other side, on the 3' end, after the coding region, you have this 3' UTR. Now 3' UTR is located just after the stop codon, and 3' UTR is important to indicate the termination of translation process. So before the coding sequence, we have 5' UTR, which indicates the initiation of translation, and after the coding region, we have 3' UTR, which indicates the termination of translation. So this is something very important to remember. Now after the 3' UTR, or at the end of 3' terminal of mRNA molecule, there is a very long sequence which consists of adenine, and apparently this can be the size, and this long sequence of adenine, it is called poly A tail, and the size of the poly A tail varies. It could be shorter or it could be very long, and the important function of poly A tail is to make sure, or is to increase the stability of mRNA, because as soon as the mRNA translocated from nucleus to cytosol, it can degrade very fast, but poly A tail can provide the stability of the mRNA molecule, and that will give the enough time for mRNA to translate this sequence into protein synthesis. So basically, poly A tail increases the stability of the mRNA molecule when it is translocated into the cytosol. And one important thing to remember is poly-A tail is a very special characteristic only for eukaryotic cells. This poly-A tail does not exist in prokaryotic cells, so this is only specific to eukaryotic cell. So now, just to quickly recap, we have this 5' cap, which is called a methyl cap, which indicates that mRNA molecule is completed and translocated into the nucleus. We have this 5' UTR, which indicates the initiation of translation process. Coding region, it encodes for protein synthesis. 3' UTR indicates the termination of translation process, and at the end on 3' terminal end of the mRNA molecule, we have a long adenine sequence, long sequence of adenine, which is called as poly-A tail, which provides the stability of the mRNA molecule in the cytosol, and that will give the enough information to transfer that information from the coding region of the mRNA to synthesize protein. Now, the second type of RNA is called transfer RNA. So transfer RNA represent 15% of total cellular RNA, and they are metabolically very stable compared to mRNA. And the another important thing to know about transfer RNA is they are smallest in size compared to all other RNAs. And they have extensive intra-chain base pairing. So these are intra-chain base pairing. So they are very extensively present in the transfer RNA molecule. And because of their extensive intra-chain base pairing, they form a structure which looks similar to clover leaf shape. So this is a structure which looks similar to clover leaf shape, and that structure can further. So this is a secondary structure, and this can further transfer. This can further form tertiary structure of the transfer RNA molecule. Now, the another important thing to remember about transfer RNA is it has on its 3' on 3' terminal of the transfer RNA has 3 nucleotides, that is C, C, A, and importantly on the adenine of 3' terminal, which acts as a carrier of amino acid, because that's where the amino acids bind on transfer RNA. Now, it also has this anticodon loop. This is important because anticodon has 3 nucleotide sequence. They are also called as triplet because it has this 3 nucleotides. So it contains this anticodon triplet sequence, which is basically complementary to the codon sequence of codon triplet sequence of mRNA molecule. This is important to remember, guys, that transfer RNA has anticodon triplet sequence, which is complementary to the codon triplet sequence of the mRNA molecule. And also there are 20 different amino acids, right? So it is so there are basically one transfer molecule for each 20 amino acids. So this is, suppose this is tyrosine. Suppose tyrosine is attached to adenine molecule, attached to adenine on 3' end of the transfer RNA. So what will happen that during protein synthesis, transfer RNA reads the codon triplet sequence of the mRNA molecule, and this particular codon sequence indicate for particular amino acid, because that is each amino acid has a different sequence. So this indicates one particular amino acids, and transfer RNA reads this codon triplet sequence, and accordingly it brings the anticodon sequence, which is indicated for one particular amino acid. Suppose in this case it is tyrosine, so it will be a different codon sequence for serine amino acids. So likewise, there is one particular transfer RNA for each 20 amino acids, and that is very important to remember. Now lastly, we have ribosomal RNA. So ribosomal RNA represents 80% of total cellular RNA and have a longer half-life compared to other RNAs. Now they are GC-rich RNA molecules, and they can further fold it with their base pairing. And they provide this scaffolding needed to assemble ribosomes, and we are going to look into this. We're going to talk about this shortly. And most importantly, they play a catalytic role in the formation of peptide bonds during protein synthesis, because they kind of act like an enzyme to provide the catalytic reaction. Therefore RNAs with the catalytic role, they are called as ribozyme. And one important thing to remember is RNAs are basically the site of protein synthesis, and as well as it also provides a catalytic role in the formation of a peptide bond during protein synthesis. Now, in prokaryotes, there are 3 distinct species of ribosomal RNA. They are called 23S, 16S, and 5S. And this S is nothing but it just stands for Swedberg unit, which indicates the molecular weight and shape of the molecule. So they all depending on their molecular weight. So 23S is the longer molecule, 16S, and then 5S. And in eukaryotic cell, we have 28S, 18S, 5.8S, and 5S. So these are the different species of ribosomal RNA in prokaryotic cells and eukaryotic cells. And lastly, when the ribosomal proteins are added to the ribosomal RNAs, it yields the ribosomal subunit. So this is our ribosomal subunit. So we have here large ribosomal subunit and small ribosomal subunit. So ribosomal RNA plus proteins form these different subunits. So let's talk about first eukaryotes. So in eukaryotes, the large ribosomal subunit contains 28S RNA, 5.8S, and 5S RNA plus 50 proteins, and that eventually forms the 60S ribosomal subunit that represents the large subunit of ribosome. Whereas small ribosomal subunit, which contains 18S ribosomal RNA and 33 proteins, total it forms 40S subunit of small ribosomal subunit. So all these ribosomal RNA which we saw earlier, so they all play an important role in forming this large subunit and small subunit of ribosome. And this actually is very crucial because ribosome is the site of protein synthesis. Now, in prokaryotes, we have for the large subunit, which contains 23S RNA, 5S ribosomal RNA, and 34 proteins, and total it forms 50S large ribosomal subunit. Whereas in small ribosomal subunit contains 16S ribosomal RNA and 21 proteins, and eventually form 30S ribosomal subunits. So this is just an example that when the ribosomal proteins are added to ribosomal RNA, it eventually yields the ribosomal subunit, and this is important for and this is very important because ribosomes are the site of protein synthesis. So this is just an overview of overview of DNA transcription and protein translation, and I will definitely be talking or I'll definitely be going to have a separate video on transcription and translation, so don't worry about thinking in detail how this process occurs, because I'm definitely going to have a separate video on each of these topics. So just to give an overview that from DNA, which is located in the nucleus, and the genetic information DNA is transcribed into mRNA. So this is the mRNA which is synthesized. I haven't really shown the structure of mRNA, but you just need to understand that mRNA, which has the methyl cap, then 5' UTR coding region, 3' UTR, and the poly-A tail. And once this mRNA is synthesized, it is then translocated into the cytosol, where it is then used to, and this coding region on the mRNA is used to synthesize the protein. So as I mentioned before, that this codon triplet is then read by transfer RNA. So this is the transfer RNA, which has this amino acid attached on the 3' terminal of A, and this is the transfer RNA, and which has this anti-codon triplet sequence. So transfer RNA reads this codon triplet sequence, and this codon triplet sequence code for one particular amino acid. So transfer RNA brings in that amino acids, and then that amino acids is being added into the growing peptide change and becomes the polypeptide change, and this amino acids are this peptide bond is formed by help of ribosomal RNA with the help of its catalytic role, which is called as ribozymes, which we saw before. So this is how the entire process works, and eventually forms this polypeptide chain is then forms used for it indicates the one particular protein. So this entire process is used to synthesize a protein. And don't worry about this, we are definitely going to talk about this in detail in next subsequent lessons. Now, other than 3 major important RNAs, there are other types of RNAs which also exist in small amount. So the one such RNA is called small nuclear RNA, snRNA, and this RNA is packaged with the proteins to form nuclear ribonucleoproteins, which is also called as SNRPs, and it is part of spliceosomes, important in splicing process, which we are going to talk about in our subsequent lesson, which is important for post-transcriptional modification, and it also important in exporting mature mRNA from nucleus to cytosol. Next, we also have small cytoplasmic RNA, which is again packed with the small cytoplasmic ribonucleoproteins and also called as scrapes, involved in protein processing and secretion. And we also have a ribonuclease P. They are catalytic RNA, and like I suggest that because they have a catalytic role, they are also called as ribozyme, and it is also important in RNA splicing during gene expression, as well as important for transfer RNA biosynthesis and during viral replication. And we also have here short interfering RNA. This is very important. It is short because it only has 21 to 24 they are only 21 to 24 nucleotides long, and they are short antisense transcript derived from transposons and complementary to certain mRNA. Because they are complementary to certain mRNA, it can inhibit the translocation of transfer of target mRNA and enhance their degradation. So basically, short interfering RNA, it is a very important tool which is being utilized in research regularly. So if you want to knock down, or if you want to inhibit the function of one particular protein, you can target you can use the siRNA, which is being targeted against that particular mRNA, and so it will inhibit the translocation of that particular mRNA, which encodes for the protein, and eventually it will inhibit the synthesis of one particular protein. So if you can inhibit the function of one particular protein, you can answer many questions during the research, that okay, like what is the function of that one particular protein. If you knock down, is it exacerbating the disease condition, or it is improving the function of certain cells. So siRNA is a very important tool which is being used in research regularly. Now we also have this microRNA, and they are approximately 22 nucleotides long, involved in RNA silencing and post-transcriptional regulation of gene expression. Now microRNAs are also very important because they are present in many mammalian cells, as well as they are also available as a circulatory microRNAs, and these are very important because they can also be used as a biomarker in certain disease condition. So microRNAs are also one of the tools being used in the research very regularly. And the important function of the microRNA is that it can actually cleaves the mRNA into two species, and inhibits or destabilize the microRNA. The other way it can destabilize the mRNA is by degrading their poly-A tail, which is important to stabilize, as we saw earlier. So therefore it is microRNA can also destabilize mRNA by degrading the poly-A tail. And lastly, we have this mitochondrial RNA, which contains their own protein synthesizing machinery, and it expresses unique RNA species such as 16S and 12S ribosomal RNA, 22 transfer RNAs, and 13 messenger RNAs. So these are some of the different types of RNAs which exist in small amount, and I will be talking about the role of these RNAs as we go along in the next subsequent lessons of transcription and translation. So thank you so much for watching this video, guys, and I hope you learn something from this video, and if you do, please like and share the video and subscribe the channel. Thanks so much for watching, and I'll see you next time.